In this session, we will be discussing stroke, particularly an old infarct. We will be examining a specimen obtained from a 62-year-old man who had experienced a sudden onset of left-sided weakness and sensory deficit 3 years previously. A residual left hemiparesis affected his arm more than his leg. He later died of metastatic colonic carcinoma. Macroscopically, we have a coronal slice sectioned through the cerebrum at the level of the thalamus. Here we can see a large destructive lesion in the region of the right sylvian fissure, this area here. This lesion involves the parenchyma from the cortical surface of the brain, here, through to the lateral ventricle, here. We can also see that the right ventricle has undergone dilatation secondary to the loss of cerebral tissue. At the macroscopic level, we would expect to see foamy macrophages and fibrous astrocytes.
Hello, we're now going to show you some of the microscopic features of an old infarct. In this case, it's a small infarct that has occurred in the cortex of an individual, and this infarct probably happened around at least 6 months before the person eventually came to post-mortem. So it wasn't responsible for killing the individual. Having a look at a low power view, what we can see here is normal brain. Then there is a sulcus, which is separating two gyri. The sulcus contains blood vessels, which will be in the subarachnoid space. And then we see on the opposite side to the normal brain this large cavity that seems to contain a large amount of cellular debris. Now, if we go up onto high power on the normal brain, hopefully you can see some characteristic pyramidal shaped neurons down here in layer 3, and they have got their apical dendrite pointing up to the cortical surface. If we then move across to the cavity or the infarct itself, there's an interesting area on the cortical surface or next to the cortical surface that seems to be spared from destruction, and we refer to this area as subpial sparing, and that's because that this area is close to the cerebral spinal fluid and of course it's in the subarachnoid space and close to these vessels, and so can still be supplied with nutrients. But although it's still alive, this particular area isn't functional, and rather than being normal, what we see in this area is that the astrocytes in particular have quite a strange look about them. If you remember astrocytes from normal brain, we could identify them as a medium-sized nucleus with a diffuse pattern of chromatin. In this area of subpial sparing, and also on the opposite side of the cavity of this particular infarct, the astrocytes have taken on a quite different shape, where their nucleus is now surrounded by a considerable amount of cytoplasm. We refer to this as reactive astrocytes, and in fact the term astro or astrocyte star is really referring to this type of astrocyte. And what these astrocytes are doing, by upregulating, they are warding off the damaged area of the brain and the infarct from normal brain. The other thing that I'd like to show you is what's happening in the cavity itself. And as I said, the major part of the cavity it contains is just debris. We don't really identify any of the classic cells that we would see in normal brain. But one cell that we can see is various sizes of macrophages, and particularly a very large one here. Now, these macrophages are called foamy macrophages, but unlike those that you'll be familiar with atherosclerosis out in blood vessels, here it's not cholesterol that the macrophages have picked up, but it's actually the myelin, the stuff made by oligodendrocytes that insulate the fibers and allow fast conduction speeds. So these macrophages or foamy macrophages is one of the few cells that we would still be able to recognize within the middle of this cavity of an old or remote infarct.